Okay, today we're going to start cell respiration, part 4. It's going to be the prep reaction and Krebs cycle. We're finally moving into the mitochondria, powerhouse of the cell. This is where a lot of ATP synthesis occurs. Far more ATP is made here than how much ATP is made without the mitochondria or without oxygen. So earlier in this unit, I've done this drawing and I'm going to do it again. This right here is a cell. This is the mitochondria. And within the mitochondria, you have cellular respiration. I've asked you to associate the word respiration with oxygen. That means that this environment is aerobic. Oxygen is required. What we've been doing last Wednesday and Thursday for cell respiration part 2 and part 3 was glycolysis and fermentation. They both take place in the cytoplasm of a cell. Cytoplasm is an anaerobic environment. No oxygen is present. And the types of processes that actually happen here are glycolysis and fermentation. So in the event that there is oxygen present, so I'm going to put O2 with a green check. There is oxygen present. The products of glycolysis are going to proceed into the mitochondria, and you're going to perform cell respiration. In the event that oxygen is not present, put O2 with the red X. You're going to proceed to fermentation, which takes place in the cell, or in the cytoplasm of the cell, not in the mitochondria. You only make 2 ATP here. But if you were to go into the mitochondria, you would make 36. Big difference. Big difference. Or actually 38 if you include glycolysis. So we're finally getting into the mitochondria today. So a few things to understand before we actually get into that. The chemical equation for cell respiration is as follows. What is C6H12O6? That's glucose, of course. How about O2? CO2? And H2O? And what is ATP used as? Adenosine triphosphate is energy. All right. So when it comes to when it comes to cell respiration, it is an aerobic process. There's 2 ways to describe aerobic. Either 1, it requires oxygen. Another way you can say this is it occurs in the presence of oxygen. And it obviously takes place within the mitochondria. Now, back when we started this whole unit, Unit 3, Cellular Energetics, I diagrammed what the different parts of a mitochondria are. I'm going to do that again with you guys right now. So a mitochondria can be circular, it can look like a sausage or a bean. And the first part that you see right here is the outer membrane. Here you've got the inner membrane, but it actually goes by another name. But we are going to call it the inner membrane. We're also going to call it the cristae. When we get to the electron transport chain with the next lesson, you're going to find out that the electron transport chain takes place in the cristae. All these little folds that are pushing into the interior of the mitochondria, that is made up of the inner membrane. But that design and the actual system that is embedded in that membrane is a whole nother part of the mitochondria that we're going to refer to as the cristae as well. So inner membrane, cristae is really going to be the same thing. And then the space between the outer membrane and the inner membrane is pretty easy. It's called the intermembrane space. And then you have this area I'm coloring in blue. This region is called the matrix. The matrix is where the two processes that we are going over today take place. It's a big metabolic pool. This is where a lot of metabolism within the mitochondria occurs. It's not necessarily where energy is made, but it's where what you need to make energy is made, if that makes sense. So you have the outer membrane, the inner membrane called the cristae, the intermembrane space, and the matrix. So let's go over the stages of cell respiration. Remember that this all begins with glycolysis, so I'm still going to mention that these follow glycolysis. Okay, first, we have the preparatory reaction. You can call the PrEP reaction for short. This takes place within the matrix of the mitochondria. Following the PrEP reaction, we have the citric acid cycle. This is called the Krebs cycle as well. And this is in the matrix of the mitochondria. And then lastly, what we'll get into for the next lesson is the electron transport chain. can call it the ETC. This takes place in the cristae, which is the inner membrane of the mitochondria. Okay, guys, I want to make something clear to all of you. Depending on who you ask, some people like to make the prep reaction part of the Krebs cycle. Some people don't. I'm going to make them separate, but just in the event that it happens, I want to make this disclaimer for you. Some people consider these two stages to be one in the same. So there will be times where you will see, it may seem as if the prep reaction was completely skipped. When it wasn't really skipped, it just means that they wanted to make it part of the citric acid cycle. But the reason why it's called the prep reaction is you are preparing for the Krebs cycle. So we're going to distinctly separate the two of them so you know what's happening in the prep reaction and how that leads to the citric acid cycle. And then for the electron transport chain, this is where the bulk of ATP is made. And we will get into that for the next lesson. Like a lot of ATP, 34 molecules of it, at least. Okay, now, you all see some stuff written on the whiteboard, but for the sake of the people watching this video, I'm going to write what you see on the whiteboard on the screen here. And this is going to be your 2 electron carriers. Electron carriers ferry electrons to the electron transport chain. That's all they do. Uber Eats ferries food to your house. The Staten Island Ferry in New York ferries people from Staten Island to Manhattan. Uber will ferry you from Busch Gardens to your house if you don't have a car. I don't know. It's just a means of transport. And the 2 electron carriers that we've done, well, one of them you know. NAD plus will become NADH. I want to go over again how this is actually formed. So first, you start off with NAD plus. And then you add a hydrogen ion to it. You'll get NADH with a positive 2 charge. You then add an electron to that, and you'll get NADH, a positive 1 charge. The charge went down a little bit. You then add a second electron, and you get NADH, and it has no charge. It is neutral. Anybody tell me how many coupled reactions are on this? How many different reactions are working together to form like one big process? What do you see? 3. You got reaction A, reaction B, reaction C. To help you see it perhaps more clearly, there's 1, there's 2, there's 3. The products of reaction A is a reactant to product B. The products of reaction B are reactants to reaction C. You've seen NAD plus a lot. You're going to see it a lot today, but you're also going to see a new one today, and that is going to be FAD to FADH2. You only see this 1 electron carrier once, and you see it in the Krebs cycle. So I want to show you how this goes from FAD to FADH2. Because this is new, I'm going to tell you what's included in it. It carries 2 electrons, and it carries 2 hydrogen ions. NAD plus can carry 2 electrons and 1 hydrogen. FAD can carry 2 of each. So here we go. you've got FAD, it grabs an electron, or excuse me, it grabs a hydrogen ion, and it will become FADH with a positive 1 charge. This is what you can call reaction A. You then add a second hydrogen ion to it, and it will be FADH2 with a positive 2 charge. This will be reaction B. Moving on, you then add 1 electron to it, and its charge will go down by 1. It's now FADH2 with a positive 1 charge. This is going to be reaction C. And then lastly, you add 1 more electron to it, and it's going to be FADH2 with no charge. It is neutral. This will be reaction D. Now, if you're sitting at your desk like, what is he talking about? Reaction A, B, C, D. Think of it like this. When you took freshman biology, honors bio for most of you, you went through the whole year, and at the end of the year, you learned all this stuff about cells and ecology and evolution and genetics. You learned all this stuff. By the end of the year, that was your product. All the new stuff that you developed. Well, it turns out that for this class, AP Biology, what was once your product is now your reactant. You need all that stuff you learned freshman year to be able to understand this stuff. So that's what we call a coupled reaction, where the products of reaction A are now the reactants to reaction B. The products of reaction B are the reactants to reaction C, and the products of reaction C are now the products of reaction D. You just keep building and building and building. That's what coupled reactions are all about. So this is your second electron carrier. You're going to see this when we get to the Krebs cycle. But without any further delay, let's go ahead and get into diagramming these things. We're going to do the prep reaction first. This one's actually very brief. It should take us less than 5 minutes. It's actually very simple. Does anybody need the stuff I have in orange still? We're good? All right, let's do this. Now, I want you all to look back at your notes from glycolysis, which was, I believe, part 2 of this entire lesson of cell respiration. And I just would like someone to tell me what were the end products of glycolysis? When it was all said and done, what are the carbon molecules that we ended with? We went through G3P, BPG, 3PG, PEP, and then what was it turned into? Pyruvate. How many molecules of pyruvate? 2. Here's my disclaimer for all of you. I do not have time to draw double everything today. So what I'm going to say is everything that we draw is double. So when you are all finished with this drawing, you must realize that everything is actually in quantities of twice as much as what you see. All right, here we go. This is pyruvate, your C3 molecule. This is what you ended with in glycolysis. Well, coming into the picture is going to be NAD+. And that will result in the formation of NADH. I'd like to ask you as a class, what is the word where electrons are taken from a molecule? Just like what NAD+ did to pyruvate. It was oxidized. So here's how you would say it. You would say NAD+ oxidized pyruvate. But then flip that. What did pyruvate do to NAD+? It reduced it. So you have to look at it like the Buccaneers beat the Packers by 28. And what's the other way of saying that? The Packers lost to the Bucks by 28. You have to look at it from both sides. So you can say, all right, NAD plus took 2 electrons. You could say NAD plus oxidized pyruvate. Or you could say pyruvate reduced NAD plus. You have to look at it from both perspectives. So this is also going to cause a carbon molecule to come off in the form of carbon dioxide. So that's 1 carbon. You only have 2 left now. This is going to form something called an acetyl group. You could just call it acetyl. It is a C2 molecule. Now, lurking over here is a coenzyme. We're going to call this coenzyme A. You can call it CoA for short. It is going to bond with this acetyl group to form something called acetyl coenzyme A. And after that, we're going to get into the Krebs cycle. Which we'll draw momentarily. But that's it. That's the entire prep reaction. You basically just catabolize pyruvate from a C3 molecule to a C2 molecule. You got rid of 1 carbon dioxide and you made a couple more, or 2 carbon dioxides, you made a couple of NADHs as well. Now, just like on the FRQ portion of your test, drawings will not suffice, so we need to actually have some explanation as to what is going on here. So I'll let you guys finish up, and I'm going to add some actual written notes to this before we get into the big Krebs cycle. Ready? All right, so the pyruvate formed from glycolysis enters the prep reaction in the mitochondria. Two NAD plus molecules are reduced to NADH, and they're going to, oops, that's not a very good parenthesis. They go to the electron transport chain. Then lastly, coenzyme A binds with acetyl, so it may proceed to the Krebs cycle. And what does our body do with the carbon dioxide that is produced in our cells? It gets rid of it. What happens is the CO2 gets into your bloodstream. Your blood will transport it to your lungs. It will diffuse into your lungs, and you will exhale it and return it to the atmosphere for autotrophs like plants. All right, you ready for the big one? Let's do it. Krebs cycle. Okay, guys, this is going to be a cycle. It's going to be a loop. Look at me. It's going to be a loop. That's what a cycle is. So it's not going to be a straight line like glycolysis was or like the prep reaction was. It's going to be a loop. Here we go. We're going to pick up where we left off with the prep reaction. This is the acetyl coenzyme A. Now, this is jumping into the Krebs cycle. What was already in the Krebs cycle was a 4-carbon molecule called O-oxyl acetate. This is a C4 molecule, meaning that there are 4 carbons. This C4 molecule is going to bind with our new C2 molecule that just jumped into the cycle. And I know you guys can do this. What is 4 plus 2? This is going to form a C6 molecule called citric acid. Citric acid also goes by the name citrate. Just like pyruvate is pyruvic acid, and lactic acid is called lactate. This is a C6 molecule. Now, after citric acid is formed, a couple things happen. Your electron carrier comes back in, and it oxidizes the citric acid cycle to form NADH. I want to remind you again that in this diagram as well, everything is doubled. If you want to write that down, everything is doubled, just as it was in the prep reaction. I don't have time to draw everything twice. Now, a carbon dioxide molecule will break off. So there goes one of your carbons. You now are going from C5 to, or excuse me, C6 to C5. This is called ketoglutarate. It's a C5 molecule. So this is catabolism. You're breaking down a high energy molecule to a low energy molecules, and as a result, you're releasing energy. All that NADH is getting collected to, which is eventually going to make a lot of ATP. Okay, let you guys catch up. There's a lot of stuff happening here after ketoglutarate is made. So let's do one at a time. Let's not get overwhelmed. First thing, there's 3 things here. First, it's being oxidized again by NAD to form NADH. That's the first thing that is happening. Second thing that is happening, another carbon dioxide molecule gets broken off. So now we're down from C5 to C4. Third, ADP is being synthesized from into ATP. So there's 3 things that happen here after the ketoglutarate has been oxidized. You now have a C4 molecule. 1, 2, 3, 4. This is called succinate. You can see we're getting close to bridging this whole cycle. We only have 2 more steps. All right, here, everybody, here's your new electron carrier, FAD. I told you, you're only going to see it once. How many electrons can it take? 2. How many hydrogen ions can it take? It's on the board. 2. You went from FAD to FADH2. You did not take any carbons, though, so still a C4 molecule. This is going to form fumarate. Now, I can kind of see it in your faces that you're a little turned off by all these weird names. All-oxyl acetate, fumarate, succinate, ketoglutarate. That's not the point that I'm trying to impress upon you. What I'm trying to show you is you have a big C6 molecule like citric acid, and it's slowly being broken down. And as it's being broken down, you're making ATP, you're making NADH, you're making FADH2. You're getting a lot of energy from the breakdown of these molecules. That's what I want you to get. If you're watching the Avengers, I don't expect you to know all 50 superheroes' names. Get the main idea. All these superheroes are working together to beat the bad guy from destroying the universe. That's it. You don't need to have everybody's name. You don't need a cast. Okay, this last step to finally complete the cycle. You have NAD plus being reduced one more time. Make NADH. That's it. That is the citric acid cycle. One thing I did not mention, I think I should. The coenzyme, guys, this orange square I drew, that leaves, it disconnects, it's no longer part of, it's done its job. That backstage pass to help it get into the actual Krebs cycle is no longer needed. So it breaks off and gets reused and goes back to the prep reaction. Okay. So I'm going to talk a little loud now. So you got acetyl coenzyme A, a C2 molecule, only 2 carbons. It is being escorted into the Krebs cycle by coenzyme A. You grab coenzyme A in the prep reaction. Once acetyl is in the Krebs cycle, this coenzyme is going to disconnect. It will go back to the prep reaction and be reused for the next acetyl that is made. And then throughout the whole process of this Krebs cycle, you go from C6 to C5 to C4, back to C6 to C5 to C4, over and over and over. You make a pair of NADHs here, here, and there. 3 different areas where you're making NADHs. You have one area where you're actually reducing FAD to FADH2, and you're even actually able to make some ATP. Let's see if you remember this. What do you call the creation of ATP that is a result from directly breaking down a high energy molecule to a low energy molecule? What's that form of ATP synthesis known as? Substrate level ATP synthesis. It's the direct formation of ATP from the breakdown of a high energy molecule to a low energy molecule. The reason I'm being so direct with that is because tomorrow you'll learn the other way in which you make ATP. You make ATP from pumps rather than from the direct metabolism of a molecule. But this is the Krebs cycle. So let's get some writing in here, and then we'll get some inputs and outputs, and then that will be it. So you have the acetyl from the prep reaction joins a C4 molecule to form citric acid. Citric acid is repeatedly catabolized to produce, well, there's a lot of things we produced. We produced CO2, which is a waste product. You produced ATP, which is cell energy. You produced NADH, which is an electron carrier. And you produced FADH2, which is another electron carrier. By the time you're all done with it, you end up with a C4 molecule again, and that C4 molecule joins up with the next acetyl group, and the whole cycle just continues over and over and over. So let's go over the inputs and the outputs. We did not do the inputs and outputs for prep reactions, so let's do that up here first. I would like you guys to remember that everything in the prep reaction diagram and the citric acid cycle diagram is doubled. So how many pyruvate entered the prep reaction? 2 pyruvate. How many NAD pluses? How many coenzyme A's? Okay, and I think that's it. Output. How many carbon dioxides were formed in the prep reaction? How many NADH electron carriers? And how many acetyl coenzyme A's? 2. So there's your prep reaction inputs and outputs. So if you combine them with glycolysis, so far you have reduced 4 NADHs. Then let's go down to the Krebs cycle and get all the inputs and outputs from this. How many acetyl coenzyme A's entered? How many NAD pluses?
6.
Good, you doubled everything. 6. How many FADs?
2.
And how many ADPs? Okay. Outputs. How many carbon dioxide molecules? 4. How many NADHs?
6.
How many FADH2s?
2.
How many ATPs?
2.
Okay. What does your body do with the carbon dioxide? It's released into the atmosphere. You exhale it. It goes to your bloodstream. It then diffuses into your lungs and out of your body. What are your cells going to do with the NADH and the FADH2? Where? No. The electron transport chain, which we will do next lesson. And what do your cells do with ATP? Used for energy. Okay, so I keep mentioning the electron transport chain, ETC. You guys are like, well, I don't know what that means. You will tomorrow. So hopefully that last little void in your understanding will be finished. Before you go, listen, here's a little study strategy. When you're studying this is not going to learn itself. You all know that by now, you're college level students. Here's a tip. If you can teach this to someone and you can clearly and coherently express and convey the idea, then you've got it. Teach it to your dog. Teach it to your plant. Teach it to your mom. And if they ask questions, and if you can't answer those questions, you have some more studying to do to get on the right track. But if you can coherently teach this to someone else and they can understand it, you should feel pretty good about your own understanding. All right, we are done.